Metabolisms and enzymes. Oh, yes, this is the good stuff. All right, so let's take a look at metabolism. Now, metabolism, when we talk about that, it's the sum of all the chemical reactions that are occurring. So all the things that are occurring, the catabolism and the anabolism, the catabolism as the breakdown of energy and the anabolism as the building of energy. And so the catabolism, remember we talked about the energy being stored in the bonds, and so when you break that bond, you're releasing the energy. And then anabolism is actually building those building those bonds. So when you're assembling molecules, you're building those bonds. So catabolism is, whoops, let me go back here. Catabolism is the we talk about it being the potential energy because it's stored in the bonds. It has the potential to be energy, but it's just stuck there right now. And we also refer to catabolism as an exergonic reaction. X means to release, and so exergonic is releasing heat to the environment. So catabolism, a good way to remember it is think catastrophe. And when there's a catastrophe, things are crazy and things are breaking up. So with catabolism, you have the release of the potential energy of the breaking of that bond, and it's releasing heat when this reaction is happening. Anabolism is the assembling of molecules that requires kinetic energy. It requires energy in order to do this. So it's going to take in heat. It's an endergonic reaction because it's absorbing heat in order for the reaction to happen. And you want to think like anabolic steroids. Anabolic steroids build muscles. They do other things too, but they build muscles. And so you're assembling molecules through building this bond. And all right, so let's look at the different types of energy. We have kinetic versus potential energy. And kinetic energy is energy of movement, and potential energy is energy at rest. So over here we have at movement, the kinetic energy, and this is at rest. It's just sitting there. So like I said before, this is like storing in the bonds. It's just sitting there waiting. When you break those bonds, that energy is released. Now, in living organisms, the chemical energy, like I said, is found in the chemical bonds. So the process of cellular respiration and digestion release the energy for use by the cells. The source of all the energy for the earth is the sun. So that process of photosynthesis allows plants to store the solar energy in the form of chemical energy, which is sugar. This is important. So the chemical energy, or the solar energy, is stored in the chemical energy of sugar. All right? And then once we make the sugar in photosynthesis, then we'll go through the process of cellular respiration, and we'll start breaking those bonds and releasing that energy. All right? Now, moving on to thermodynamics. Oh, here we go. This is just looking at potential versus kinetic energy. This is the actual structure of ATP. I don't even want you to do anything with that right now. Just identify it. Just look at it, but that's not going to be on the test for ATP. Parts of this will certainly, though, when you look at the phosphate groups and your sugar, your 5-carbon sugar here. All right, and again, potential energy in photosynthesis in this process. So light energy is absorbed in the chloroplast of the during photosynthesis, and then in the mitochondria. Remember, that's the powerhouse. So we store the energy in the form of glucose here, and then it is broken down and released. Those bonds are broken. ATP is made in the mitochondria. All right. Now with thermodynamics, this is the study of heat. So thermo means heat, and its properties, which is the dynamics. So the first law of thermodynamics is also called the principle of conservation of energy. And that is that energy can neither be created nor destroyed. It can only be transformed or transferred. And the second law of thermodynamics is that every energy transfer increases the entropy of the universe. And so entropy is the amount of disorder. So it's a measurement for disorder. And so you're unable to do work because it's in this low state of order. So sunlight has high quality energy going in and heat, which is low quality energy coming out. So it can't do work. Okay, then we have this concept called, and you can trace this. Here's the energy flow is in red. So the energy is coming from the sun to the primary producers, and go straight to the detritus, or it can go to primary consumers, secondary consumers, and then detritus. Again, I don't want you to memorize this chart. I just want you to see it and recognize the different levels, that this energy is transferred. It's not consumed and it's not destroyed, but it is definitely transferred. All right. The making and using of ATP is this constant recycling process. So ATP is adenosine triphosphate. That means that we're talking about 3 phosphate groups. So when we're looking at adenosine triphosphate, let's just say that's our adenosine, and then we'd have 3 phosphate groups coming off of it, all right? When we break the bonds on one of those phosphates, we end up with adenosine triphosphate here, diphosphate, plus this other released phosphate group. So we end up with adenosine diphosphate, 2 phosphates here, and that bond has been broken. All right. Okay, now this free energy, it's called Gibbs free energy. That's just the scientist that figured that out. And it's represented as a capital letter G. And so it's referred to as free energy because it is available to perform work. This is mainly in your ATP or in the GTP in a cell. Not all energy is available. Some is lost as waste, like when we defecate. The same goes for cells too. So when we eat, you're not using every bit of that food. Some of it goes out as waste. Most energy is lost as heat, as a byproduct. If we could tap into all of the energy in a reaction, we'd be in great shape. All right, the types of work that are being performed, by living cells, most of them are achieved by using proteins and enzymes. So you can have mechanical work, you can have transport, you can have metabolic processes. And those are the different types of work that are being performed by living cells. Okay, so let me go back here for a moment and look at the ATP. So ATP, this is the ribose sugar and the nitrogen base adenine, and then your phosphate groups, the triphosphate. This is highly unstable, like a compressed spring. Unstable means it has the capacity to do performance work. Now ATP, like I said, converting to ADP gives off energy. That bond is broken, it's released. There's a process called phosphorylation where we put the phosphorus in here. So it's the attaching of this unstable phosphorus group, phosphorus ion, to another molecule and making it unstable and thereby able to perform work. So it's able to perform work. So here is this free phosphate ion, and it's going to attach here to the adenosine diphosphate to make adenosine triphosphate. All right. Okay, so a way to think about it is if you're looking at a stuffed animal to represent that third phosphate group, and so if you had work, Okay, that's just not making any sense. I don't even know why that's on there. Let's just move on. All right, so let's look at enzymes. So enzymes are biological catalysts. All right, biological, they're living, and the catalyst is going to make a reaction happen. So enzymes, of the macromolecules, enzymes are proteins. And what they do is they speed up and control the rate of a chemical reaction. They're recycled, they're not consumed, so they go back through the process, and they are selective with what they work with. We think of it kind of like a lock and key model, which is an old terminology. We now say it fits like a glove, or it has an induced fit. So it's like putting on a glove and it stretches to the shape of your hand. And of course we know that enzymes usually end in ASE, ACE. So how they work is they reduce the amount of energy that's needed to start the reaction. So if it needs this much energy, in order for this reaction to happen, it's going to only need, with an enzyme, it only needs this much. So is that going to speed up the reaction? Of course it is, because it's not going to have to reach all this amount of energy. It's going to happen with this amount of energy. Think about it. If it takes Johnny 20 donuts, he's got to eat 20 donuts when he comes home from school in order to start working on his homework. That's going to take a certain amount of time to eat 20 donuts. It's going to take a while. But if he takes half the time, then if he only needs 10 donuts, then he's going to be able to start his work a lot faster. So you can think about it along those lines. All right, so enzymes have this active site. And some terminology related to enzymes is the substrate is the molecule that's being affected. Okay, so the enzyme is going to grab it and work on it, on the substrate. The active site is where the actual chemical reaction is going to take place between the enzyme and the substrate. Now this is called an induced fit model because the enzyme substrate complex, meaning there's more than one unit, so this substrate is going to fit perfectly into this enzyme, and then a reaction is going to be able to take place. The two parts are mainly held together by weak hydrogen bonds. Remember, they're weak bonds. And it's orienting the substrate molecules, and by doing this, it speeds up the rate of the reaction. Now, there's things that can affect it. Temperature, pH, salt, all of those things can affect it. There's also things that can inhibit it, and I'm going to show you those in class to show things that can affect the actual process from happening. All right, that's all we have for today, and I will go over the rest in class. Yippee!